Greetings, everyone. I'm Tayab Moin from the Department of Genetic Engineering and Biotechnology, University of Chittagong. I feel really honored to be here presenting my research in such a wonderful platform like ISCB Student Council Webinars 2021. So before starting with the presentation, I'm mourning for all of the departed souls across the world due to COVID-19 pandemic and also the people around who are affected these times across the globe. This pandemic actually has taken away millions of lives, but also augmented the significance of virus, vaccine, vaccine informatics, or other research among the general crowd all over the world. Yet, it's a matter of relief that people around the world have started having access to the vaccination programs, and hopefully, we will recover this dark period soon in the upcoming days. Today, I'm going to present our research, which is based on vaccine informatics. Since the prevalence of the virus was appeared, we have been trying to contribute to the betterment of the mankind from our side. And from that perception, we have designed a subunit vaccine against the virus using immunoinformatics. And today, without making any delay, I want to get into the topic and on how we have done it. So, as everybody knows so far, the coronavirus disease is caused by a virus known as a severe acute respiratory syndrome 2. In short form, it's called SARS-CoV-2. This disease was originated in China in December 2019. COVID-19 is a highly contagious disease that, as of August 14, 2020, it has been disseminated in around 230 countries and territories all over the world. Not only that, it has put an end to more than hundreds of thousands of lives with nearly 30 million infected cases by the time. Apace with the drug development, several pharmaceutical and educational institutions from numerous countries have demonstrated their engagement in developing COVID-19 vaccines passionately after the sequenced genome of the virus has been released officially on 11th January of 2020. However, almost all of the vaccines were still not evaluated with efficacy. In our study, a blueprint of the epitope-based vaccine was designed which might produce substantial immune response towards SARS-CoV-2. We have targeted the spike glycoprotein, nucleocapsid phosphoprotein, membrane glycoprotein, and the envelope protein of the virus. Coronavirus spike glycoproteins promote and facilitate entry into cells and they are the prime target of the antibodies. The nucleocapsid phosphoprotein is vital for packaging the viral genome into a helical ribonucleocapsid, and it plays an elementary role during viral self-assembly. Also, membrane and envelope proteins are important for viral entry, replication, and the particle assembly within the human cells. Therefore, these four proteins were used as potential targets in the study to design the vaccine with the purpose of inhibiting these viral proteins, which should interfere with the viral life cycle. So in our methods and materials and methods, we have followed the methodology starting with the selection of the virus and retrieval of the protein sequence using NCBI and UniProt database. After that, we went for antigenicity and physiochemical property analysis of the selected proteins, which was followed by the most important part of the research, the epitope prediction using IEDB server. So we have predicted T and B cell epitopes, which were rigorously assessed for antigenicity, allergenicity, toxicity, conservancy, human homology, transmembrane topology, and the cytokine-inducing ability. Finally, we have considered the best selected epitopes based on several criteria, for instance, antigenicity, non-allergenicity, non-toxicity, and conservancy of the epitopes. Well, after the CV vaccine is constructed using linkers and adjuvant, it was gone through physiochemical property analysis again, secondary and tertiary, and then the secondary and tertiary structure prediction, and finally the validation and refinement of the tertiary structure. Following that, molecular docking, molecular dynamic simulation, and the immune simulation studies were also conducted. After the prodigious analysis exploring the interaction between the vaccine and the viral proteins, the vaccine was finally subjected to the codon adaptation and in silico cloning. Well, in our results and discussion, at first, the four proteins, as I have mentioned above, were selected, and then the protein sequences were retrieved from the NCBI database. Here in this table lists the GenBank accession IDs either of the selected protein sequences. Well, the vaccine has been constructed using the best selected epitopes, as I have mentioned previously, which could be used to fight against the selected viral strains effectively. So beta defensin 3 was used as adjuvant, and the PADRI sequence was also used for the vaccine construction to increase its efficacy. So 3 linkers were also used at their appropriate positions during conjugating the epitopes to form the vaccine. The CV vaccine was predicted to be a potent antigen as well as a non-allergen. In the physiochemical property analysis of the vaccine, a high theoretical PI, a good half-life, and a good half-life of the vaccine in E. coli culture system were predicted. Moreover, the vaccine construct was also found to be quite stable as well as soluble upon overexpression in E. coli cell culture system. Furthermore, it had an aliphatic index of 114.48 and a quite low gravity value of -0.884. Then the most interesting part of the research, the molecular docking analysis was conducted, which wound up with an impressive results, yielding high free energy with a very strong interaction between the toll-like receptors of the host cell and the vaccine construct. Here in this figure, the interactions between the vaccines and TLRs are clearly shown with the interacting atoms colored in purple. After the successful docking, simulation study resulted with some efficient evidences that proved the potentiality of the virus. MD simulation was conducted on the vaccine TLR docked complexes, and MD simulation of the protein complex, which pointed towards the fact that all the docked structure were quite stable throughout the experiment. Here, the graph shows the result of the MD simulation of the CV vaccine TLR H complex. Here in this in the graph A, the RMSD graph, it shows that the structure had maintained a stable of 0.5 nanometer deviation with minimum fluctuations. And in the graph B, it showed that the RMS fluctuations of all the atoms about its average position. The peaks and the dips in the graph denote the flexibility of the corresponding region in the molecular structure. Finally, the graph C shows the radius of guidation. The immune simulation study predicted that the the SARS-CoV vaccine might produce an immune response which is consistent with the typical and natural immune system. The study revealed that after injections, the primary immune response against the vaccine was predicted to stimulate significantly, as indicated by the gradual increase in the levels of different immunoglobulins that are shown in the figure. Here, the graph A shows the immunoglobulin and immunocomplex response to the CV vaccine inoculation by the black line, and specific subclasses are indicated by the colored lines. The graph B shows the rise in the B cell population over the course of the 3 injections. Graph C indicates the increment in the B cell population per state over the course of vaccination. Graph D demonstrates the increase in the plasma B population over the course of the injections, and graph E depicts the enhancement of the helper T cell population over the course of the 3 injections. The graph A finally shows the increment in the helper T cell population per state over the course of the vaccination. And here, graph G illustrates the elevation in the regulatory T lymphocyte over the course of the 3 injections. And graph H shows the increment in the cytotoxic T lymphocyte population over the course. Graph I displays the increase in the active cytotoxic T lymphocyte population per state over the course. And graph J shows the rise in the active dendritic cell population per state over the course. And K demonstrates the increment in the macrophage population per state over the course. And finally, graph L shows the augmentation in the concentration of different types of cytokines over the course. And finally, in the immune simulation, we have observed a gradual increase in all the analysis. So for in silico cloning and plasmid construction, at first, the protein sequence of the CV vaccine was adapted by the Jacket server, and then the codon adaptation index was found to be 0.903, which indicates that the DNA sequence contained a higher population of the codons that are most likely to be used in the target organism, the E. coli strain K12 that were used. for the efficient production of the CV vaccine. Also, a good GC content of 56.79% was also recorded for the adapted sequence. After codon adaptation, the predicted DNA sequence of CV vaccine was inserted into the peptide vector plasmid as seen over here. So, therefore, the study revealed the vaccine construct as a potential candidate. Nevertheless, the vaccine construct must undergo wet lab-based inception for regulating its safety, usefulness, and competency. We hope that inhibition of the selected proteins of the virus would consequently halt the viral entry, and thus the life cycle of the virus will be prohibited. That's all for today. Thank you so much for having patience in hearing to my research talk. Thank you.